Hello everybody, my name is Justin Anair. I'm a third-year PhD candidate in the Department of Neuroscience here at Northwestern, and I'm here to talk to you about all things dopamine. So you might have heard of dopamine in popular culture. It tends to influence many types of human and animal behavior, most notably reward. And so an example of this is after like a long day of work, you're walking home from work and you start to smell like a good smell coming from one of your favorite restaurants. For me, this is Small Cheval here in Chicago. And you think, you know what, I deserve a little reward after a long day's work. So you stop in, you grab your food, and you take that initial bite and you start to feel some pleasure, and that's dopamine at work signaling that reward for you. And now dopamine also influences many other types of behavior, such as movement, learning, emotion, perception, as well as social interaction. And now dopamine is released in a lot of different parts of the brain, but dopamine release in a central region called the striatum is very important in regulating all these different functions. And so I'd love to talk to you all about these functions in the healthy human. What I'm actually here to talk to you all about is the dark side of dopamine. And so people don't realize that dopamine is actually involved in many neuropsychiatric disorders, and especially a dopamine release in this region called the striatum. So too little dopamine release in the striatum is associated with Parkinson's disease, where patients have a hard time making fluid movements. And then too much dopamine is associated with psychosis in patients. And my research here at Northwestern focuses on psychosis and studying how excess dopamine in the striatum leads to this behavioral state. And so what exactly is psychosis? Psychosis is a set of symptoms like hallucinations, delusions, hyperactive movements, as well as disorganized thoughts. And psychosis takes place in many different neuropsychiatric disorders like major depressive disorder, PTSD, postpartum depression, as well as schizophrenia. And actually, psychosis is a major factor in detecting and diagnosing patients with schizophrenia, and this is due to the presentation of hallucinations which patients can subjectively report to their clinicians. In addition, psychosis is also the major tool that researchers will use to come up with new therapies for schizophrenia due to the debilitating nature of psychosis. So how exactly can we study psychosis? So in humans, you'll know that they're experiencing psychosis in patients with schizophrenia specifically when they start hallucinating. So they'll be subjectively reporting to a clinician that they're sensing some sort of stimuli, and the clinician can tell that nothing is actually there. And so we don't have a lot of tools to be able to study in depth into the brain what is actually going on during hallucinations. And so we use animal models like mice, where we have a wider range of tools that we can ask more complicated questions. And so ideally, you'd want to ask a mouse if it's hallucinating or not, but you might think that mice don't really hallucinate. And so for the last few decades, instead of using hallucinations in mice, researchers have used running speed as a proxy for if a mouse is experiencing psychosis, and also using how well drugs are able to reduce the running speed of mice as a proxy for if they will be successful in treating psychosis in human patients. So you might be thinking to yourself, like, is running speed really that good of a output for, or like a measurement for psychosis? And ideally, it hasn't been, or it really hasn't been for the last few decades, and it hasn't been very fruitful in producing new drugs that are more effectively treating schizophrenia. And so what if instead you could ask a mouse if it was hallucinating, and it could subjectively report to you, I am hallucinating, I'm experiencing something that's not actually there. And I'm here to tell you that you can. And so this is what my research uses. And so this was developed by a team of scientists at WashU, where basically we can ask mice if they're hallucinating or not, or having hallucination-like events, and they will tell us. And the way this happens is we place mice into a chamber that has 3 ports that they can poke their nose into. When the mouse pokes its nose into the center port, it initiates one of 2 trial types. Either a sound trial, where a sound stimulus is presented to the mouse. The mouse learns to associate that sound with the bottom port here in order to obtain a water reward. And then there's the no sound trial. So no sound is presented to the mouse. The mouse learns that it needs to poke into the top port in order to receive a water reward. And so you can start to think that on no signal trials, when the mice, instead of reporting that they heard no signal, report that they heard a signal, and they report it with confidence, that we can start to interpret this as a hallucination-like event, that the mice are experiencing, or they experienced something was there when in reality it was not. And so I use this hallucination-like behavior, and I combine it with genetically engineered mice where I can increase levels of dopamine into the striatum to see how increases of dopamine in the striatum, similar to human patients, will affect this type of behavior we see in mice. And so when I combine this set of tools together, and I look at the percent of hallucinations that the mice experience during this task, I find that in red, when we increase the levels of dopamine to the striatum, you see that there's an increase in the propensity for the mouse to hallucinate. And then when we pre-treat the mice with haloperidol, which is an antipsychotic drug that's been proven to work in humans to some degree, you can see that pre-treatment of haloperidol negates the effects of this excess dopamine shown here in purple. And so showing that this type of behavior and this type of set of tools are accurate to a higher degree at predicting both hallucinations as well as how well the antipsychotics might work. And so what exactly can we use this model and behavior for? So there's really two major arms. One is we can use this for drug discovery, so we can more effectively and maybe more accurately screen for new drugs for the treatment of schizophrenia by seeing how they reduce the hallucination rate of animals in this task. And we can also ask more complex questions about the brain circuits and the cell types that are underlying hallucinations to really understand the basic neuroscience of what's going on to cause these hallucinations. And so ultimately, this latter question is what I'm interested here in Northwestern. And I want to know, how does excess dopamine influence the neural circuits and cell types in these circuits to cause hallucination-like behaviors, and hope that we can have better treatments for schizophrenia. And with that, thank you for your time, and thank you to my lab.